We are very happy to have you here, Keith.
Thank you, Aaron.
And now we'll talk about forces.
Okay, so again, truth in advertising, there's not much about nanophotonics here. There's a little bit of evanescent waves and not much optics, but there's some, we're changing gears to look at more mechanical phenomena. I'm associated both with the Cornell Center for Materials Research, which is an NSF-funded MRSEC, as well as the Department of Materials Science and Engineering. The AFM work was primarily done by Adam Pranda as a senior thesis project. He's going on at the University of Maryland and in the PhD program there. Jipeng Shi did a lot of the data analysis this summer, and Phil Karubia is on the staff and helped with a lot of the instrument design and measurements. So I want to show you an image of a push-me-pull-you. You can get this off of Wikipedia, so it must exist. For those of you who are fond of older literature, Hugh Lothing's Dr. Dolittle invented this creature which has two heads, and it has a problem that when it tries to move, both heads go in opposite directions, which is, I think, the problem often material scientists and biologists have when they're using AFM. They don't know whether to push or to pull. Sometimes it's good to push, sometimes it's good to pull. And so the push-me-pull-you has some lessons for us there. If we can talk to each other, we can go in the right direction. Also, since I'm the only thing between you and lunch, I kind of wish we were all push-me-pull-yous, because its other advantage is that it can talk while it eats at the same time. So if we were able to do that, we wouldn't actually have to have a lunch break. So that's the push-me-pull-you. This talk is really focused on instrumentation, primarily because I'm interested in bringing in new characterization techniques into the CCMR. We have a large suite of shared facilities. Our job is to offer cutting-edge technology to our users, and obviously the Nanonics tools fit right into that. So we've had a Nanonics tool in our center now for almost a decade. It's hard to believe it's been that long. MV1000, MV2000. It's been used for some NSOM measurements out of Mikhail Lipson's group. We've used it also for combined AFM Raman, looking at microfabricated graphene structures on silicon carbide. But as Aaron mentioned, we're very interested in measuring very small forces that the tuning fork affords. And if you can get down to the piconewton regime, there's really lots of opportunities for measuring forces on soft materials. And those materials could be synthetic ones, they could be biological ones. And I've stolen some slides from Nanonics, I hope you don't mind. But the Nanonics has been thinking about this for a long time, about the problems associated with measuring forces and imaging in fluid, for instance, on biological specimens. Primarily, the problems there are partly geometric. When you've got a tube scanner and you've got a little AFM cantilever in there, it's hard to do good optical microscopy as well as do the AFM measurements. But there's also problems with the cantilever themselves, because if you want to look at biological material where the forces involved are very small, you have to use very soft cantilevers. These cantilevers in fluid are very difficult to adjust. They drift. They can actually change chemically in the fluid. So there's some challenges there. This kind of illustrates what happens in a conventional AFM, where you're using beam bounce techniques, you're reflecting a laser light off the back of the AFM. As you approach the cantilever towards the surface, there's no force experience, but then there's an instability where the cantilever, soft cantilevers bend, they snap in, and then they touch the surface, and there's a repulsive regime. And then when you pull it going in the other direction, you pull it out, then there could be strong adhesion, and there can be some oscillation of the cantilever depending upon the conditions and the force constant of the cantilever. So this jump to contact, the time scale associated with measurements, the noise that's associated with these cantilevers, the soft cantilevers, are problems. And so Nanonics has proposed using the tuning fork cantilevers for measuring soft materials. They have very high force constants. In fact, if you just attach a straight cantilever at the end of the tuning fork, what's really involved is the force constant of the tuning fork itself. That's really going to give you the force information. How do you actually get out small forces out of something with such high Ks? The idea is that these have high Qs, so you can use dynamic measurements to extract that information. There's no jump to contact, no ringing. The damping in fluids is limited. So ultimately, you hope that you could use this approach to give you sensitivity for forces. So now I'd like to turn to actually a measurement that I was involved in at Cornell that highlights some of the problems of conventional cantilevers, but also the very interesting experiment, which I think will suggest opportunities for the tuning fork. This is work done on a conventional beam-bounced AFM. It's a Pico Plus from Agilent, mounted on a total internal reflection fluorescence microscope. So we're using a Nikon TI. And this is out of the group of Manfred Lindau, and the grad student was Mark Harris, and I assisted sort of with the instrumentation part. The idea here is that there are vesicles that are involved in lots of interesting cellular mechanisms, and what you'd like to know about is what's the interaction between the vesicle and the plasma membrane of the cell. As a vesicle approaches the membrane, before there's actually a fusion and transfer of material through the membrane, there's a tethering stage. And this tethering stage is something that's very interesting because you'd like to understand the mechanics of that tether. So what the aim of this experiment was to take cells where we could have a plasma membrane, observe the vesicles on the membrane, and then try to pull the vesicle off or pull on the tether. And I'll go through this relatively quickly, but the idea here is that in the turf we can see these are PC12 cells that have been sonicated. We can see the membranes that are left over from the sonicated cells. With one dye or the other dye, the vesicles are expressing this GFP. So the green, you can actually pick out the individual vesicles on the membranes. So what we're going to do is bring down the tip to the individual membranes and try and touch a vesicle and hopefully pull it off. And this shows the sort of experiment all summarized. This is the cantilever deflection, so a conventional soft cantilever. So the deflection is indicative of the force that's involved. All right. We're doing movies on an and/or camera actually as we do the force measurements. So that these are all synchronized in time. The green trace is the fluorescence in a certain area associated with what we think is a vesicle as it moves into the surface and away from the surface and into the surface. So over time, what's happening is you push the tip in and you wait. So you're pushing the tip in and you're hoping that you'll grab the vesicle, at least stick to the surface, and then you begin to pull it out. So that's what these traces are here. You're pulling the tip out, and it's called a force clamp because you're applying a certain fixed force to the surface, and then you're watching for these quick events here, these small peaks that are associated with something happening between the tip and the surface, which is postulated to be some detethering as complexes extend under that force. Now, the blue trace is the servo on the FM. Essentially, what's happening here is that you're trying to maintain the tip at a constant force, so it's bending like this. And but when there's a little detethering, the tip bends, and so you have to adjust the servo to pull this up to keep the force constant. So this is what the servo is doing: is that as the clamp is changed and there's events that change the force between the tip and the surface, you have to adjust the servo to keep the force constant. So the point of these measurements is that you can actually then monitor these sudden changes in force and associate them with changes in the vesicle position or in the tethering mechanics. And just to show you that this is where the near field optics comes in. The turf trace here is of the signal as we pull the tip out away from the surface, and what it looks like is that you see a correlation between the motion of the Z servo as the tip is moved up and also the position of the fluorescence, okay, so the associated position of the vesicle. So there's an actual correlation in the vertical distance here between the AFM and the servo, the AFM and the fluorescence. Now, so the point of these measurements is to try to understand, right, what's the origin of these force events, so you can analyze the decay rate. So if you shorten the time scale here, you can analyze the decay rate. You can watch the servo extension, understand the change in force associated with these events, and the analysis indicates that the mean length of the extension events is observed over many measurements, about 7 nanometers. This is consistent with extension lengths of unfolding of a single alpha helix complex that you might think is tethering the vesicle. So there's a lot of interesting biophysics here. I'm not the expert in that, so don't press me too hard on it. But but what you can see here is that there's some problems with this measurement because you're using this soft cantilever. There's the time decay here is associated with the fact that you've got feedback going on. So you might be losing information here because of the time scale that's available to you associated with this soft cantilever. So this is an opportunity for these kind of tuning fork measurements. Also, the adjustment of the AFM here is very difficult to do. You can imagine we're trying to get an AFM close to this membrane and fluid while the cells are bleaching. It's a very high stress experiment. So now I'd like to turn to kind of this intersection between material science and biology, there's lots of opportunities for measuring small forces in small localized regions. There's dynamic events, as I've shown you, protein unfolding, vesicle detethering, but also there's static properties that you'd like to learn about, kind of the modulus of tissue and fibers. So I'm going to now kind of simplify the whole picture for you, because that's where we are. Modulus of elasticity means the stiffness of the material in response to an applied force. In the Hertzian model, all you're doing is you're taking a sphere and you're pushing on this elastic medium, and you're trying to relate the force you apply to the modulus. And it turns out this is the equation that does that most simply. The force depends on the third power of the displacement of the third house power with the rays of curvature in there. So that's a Hertzian kind of picture. Nanoindentation is a very active field in material science. The idea there is that you like to extract mechanical properties from force displacement data as you indent a nanoscale tip into a surface. And there's many phenomena that can go on. If you're looking at a metal, there's dislocations and all sorts of defects that are involved. There can be plastic effects. In a sort of general representation, Oliver Farr described kind of a loading and unloading sequence here, where this is the displacement of the nanoindenter into the material. So this is the force, right? And so you push in, then you pull out, and there may be some difference in the displacement as you pull out because of plastic effects. What you do is you measure the slope of the unloading curve, and you can relate that slope for a spherical indenter, again, to some height function here, as well as the rays of curvature. So here's the modulus. So the point of these kind of measurements is that you'd like to do the nanoindentation and then pull out a modulus to learn about the material. So this kind of measurement now has become pretty standard with atomic force microscopes. And so I'm wearing my instrumentation hat. We have one of these Asylum MFP3D instruments in our facility, and one group at Cornell is looking at mouse aorta. All right, they're interested in the mechanical properties of this tissue. All right, so they take a standard AFM cantilever, put a bead on the end of it, and they bring it into the vicinity of the aorta. So there's no force initially. Then they push into it, and the repulsive force increases as you elastically deform that. As you pull it out, then the blue trace, there's some adhesion. All right, and this is a conventional AFM. Adhesion, and then it goes to 0. All right, and this, if you look at the software of this system, what you see is that they've already built in, all right, all these different models, right, of indentation, all right, and they give you this whole kind of suite of choices about how to fit this data, how to analyze the data. We'd like to actually then take this concept of measuring the elastic properties of material and use the Nanonics tuning fork to do that. The challenge, of course, is that the tuning fork is, the force that you get out of it is a little bit more complicated than just the deflection of the cantilever, right, which is very simple conceptually. Here you need to use an algorithm that takes into account the frequency shift, right, of the tuning fork as you bring it in to the surface, and folded into that, of course, is the spring constant, in this case, of the tuning fork. That's the mechanical component that's vibrating, right? There's the amplitude of the probe tip, and so when you use this algorithm, which is supplied by Nanonics, you can in principle extract a force and get the force distance curve. We would like to see how well the forces that you measure on a kind of standard material with Nanonics relate to other techniques. So our approach was to take PDMS, which is a conventional viscoelastic silicone, which is in a variety of materials, including chicken McNuggets, which if you stay for dinner tonight, you might want to avoid McDonald's. But you can adjust the modulus with the mixing chemistry, so you can get sort of relatively hard, relatively soft materials. But the challenge with PDMS, although it's very easy to work with and can give you this range of moduli, is that it has some nonlinear behavior. So this is actually a disk of PDMS that's been compressed between two plates in a conventional DMA. This was run by TA Instruments for us. All right, you can see that at very low loading, all right, the slope is low here, so the modulus is low, but then as the loading increases, right, we're up here at tens of newtons, that the slope gets steeper and steeper, so you're getting a kind of stiffening of the material under the loading. So that was our sample material, and what we then did was we took 3 different techniques. Our MV2000, we would love to have a multi-probe system, but we don't have one right now. MV2000 with a glass probe attached to the tuning fork. We're using very short non-cantilevered probes. So these are probes that barely stick beyond the bottom tine, the tuning fork. They're very hard to see, actually. 20 nanometer radius of curvature on these. Then we use a system, a home-built micromechanical analyzer, uses a commercial MEMS force sensor. It has a much larger probe. It's a 50 micron radius fused quartz bead that we glued to the end of it. So we're getting sort of millinewton forces here, getting kind of micronewton forces here, and then we can get up in the Newton range using a commercial tool. Essentially, this is a commercial DMA. You may be familiar with these. They're often used for looking at the oscillatory response of materials mechanically. So two plates that are controlled by non-contact electric motors that kind of oscillate back and forth. The force is very well defined. If you put a sphere on one of those plates, you actually get a little nano indenter, or that little indenter with a big radius of curvature. So we're going to use that as our sort of third technique. And after we did the AFM measurements with the Nanonics tool, we actually put it in SEM. You can see the radius of curvature is about 20 nanometers. So we're sure of that. That's very important for fitting the data. The micromechanical analyzer, this is, as I said, it's homemade. Essentially, we have these capacitive load sensors that are mounted on piezo positioning elements. This is made by a company called Femto Tools in Switzerland. There's interdigitated silicon structure here attached to your probe. At the very end of that probe, we glue our 100 micron radius bead. There's also the possibility of displacing laterally, so there's another kind of sensor that detects force sideways. You can work in fluid. We can get up to loading on the order of about 2-3 millinewtons. Okay, so here's some sample plots. This is the MMA and the DMA, MMA indentation. Sorry for the change in direction, but essentially we're pushing in this way. So this is the increasing force. And where trace and retrace are in the same color. There's a little bit of a hint of adhesive interaction here as you pull out of the surface. You can see that we're moving in only about on the order of about 20 microns. So we're not going much beyond the dimension of the bead itself. So this is not quite the Hertzian approximation where you really would only push, some fraction of the radius, but we're still close to kind of that Hertzian model. In the DMA, it's the same thing. The displacement is on the order of about 1.5 millimeters, so that the radius is about 3.6 millimeters. So again, relatively small amount of displacement. Again, the forces here are sort of tens of newtons. Here we're on the order of millinewtons. All right, so we're not seeing a whole lot we're seeing a little bit of nonlinearity here in the MMA, fair amount of nonlinearity here. There is some indication of adhesion in the DMA. So this is something we're not quite analyzed thoroughly yet, but this is what you'd expect for PDMS, often getting some adhesive effects. All right, so the Nanonics, this is just one sample trace. Again, we're coming in, no contact, and then we're going up, so blue is trace, orange is retrace. Forces are relatively low, micronewton range, and again, we're going in a small distance. We're trying to go in a distance that's sort of on the scale of the radius of curvature of the tip. We'll go a little further than that. We're going in maybe 50 nanometers into the surface. In order to analyze this data, we've had to remove some background artifacts and slope. So these are actually have been adjusted to be flat out as you move away from the sample. Remove that background slope. There's a little bit of hysteresis here. We're not quite sure the origin of that hysteresis. There are some possibilities of nonlinearities and sample drift, that's sort of associated with the instrumentation. There could be some adhesive effects. Also, the PDMS may be showing some time-dependent behavior or maybe a slight amount of plasticity. But the effect is small, all right? So we're confident that whatever we're fitting here, we don't have to worry too much about the hysteresis. So this is the results. The Albert Farm nanoindentation model, remember, is looking at sort of the slope of the loading unloading curve. What I'm showing you here essentially is a bar plot at different stress levels. Because we're using different radii curvature indenter shapes, we need to make sure that in order to compare the techniques, that we compare them to the same stress levels. Okay, so we know the forces, we know the radius of curvature, we can calculate the average stress level at certain forces, and then plot the modulus that we extract fitting only that range within that range of stress levels. We're doing fitting within a particular range of the plot. And what you see is actually very encouraging, because the AFM data, all right, is showing an increasing modulus as the stress level increases. That's what you'd expect for PDMS. The MMA is showing the same thing as is the DMA. Now the slopes are a little bit different, so you still have to understand that. But there's also very good agreement between the different techniques. So this is very different length scales, but we're still extracting very similar modules, which is encouraging, primarily for our point of view, from the Nanonics tool, because the way of measuring the force is so different than these other two techniques. And so this is a good result for us. If you do the Hertzian model, the AFM data, the moduli that we're getting are actually quite similar relative to the Albert Farr model. So here we're getting moduli in the order of 0.5 megapascal at very low stresses. Same thing here. So the model isn't making a huge amount of difference for the AFM numbers. The MMA is not fitting well to the Hertzian model, so we still have to understand that. It's not fully explained, but the DMA is doing pretty well. And again, the moduli, from the point of view of mechanical measurements, this is very good agreement, right? So the error bars here are associated with the fact that the AFM data is averaged over several measurements, and so within the error bars, these are actually identical. So in conclusion, we've taken three very different techniques for mechanical measurement that sample the modulus over different length scales, and we're getting good agreement for the moduli that we're extracting. There's some model dependence, but it's not that large. What's very important here also is that we're using the same model for each of these techniques. So however we're missing the full physics, we're making that same mistake with each technique. So it's not like we're using different models to give us the moduli. We are neglecting adhesive effects, and we have to pay a little more attention to that. But again, they're not that large, apparently, in these measurements. The AFM tip is conical in the sense that it's not a real sphere. And we are pushing a little bit beyond the Hertzian sort of simple model. So we actually would like to talk to Nanonics about how we can adapt, because they've done some work on using conical shapes to model the moduli. What we'd like to go in the future is apply these techniques to softer materials, soften up our PDMS, perhaps even look at some cell membranes, and do it in fluid, because I think that's where the real potential for this technique is. So I'll stop there and answer your question.